Welcome everyone. Today in this submodule 1.1, we'll learn how the proteins are built. We'll go through the different level of protein structures, be it primary, secondary, tertiary, quaternary, and we'll actually fetch a real sequence and spin the 3D structures inside the notebook. I'll explain both the biology and the code line by line. So even if you have never coded before, you will follow along. In this notebook, we have put together the concepts important for this notebook. And following the concepts, you will find the activities. We have put together the different sorts of practicals using PyMOL in three different ways. You can really run the practicals using the Python script, or you can run the practicals using PyMOL. Now, in PyMOL, you can either use the command line mode to run the practicals or simply use the GUI interface to run the practicals. And the last way is where you can import the PML scripts that we have created already for you to be able to run the different sorts of activities and the practicals that we'll take you through. Now, in the tutorial, my main goal is to demonstrate you the different sorts of activities in PyMOL. We'll discuss the different practicals that have been put together in the notebook. I'll skip the conceptual part. The notebooks have been created in such a way where you can go through the concepts yourself. It's a self-paced learning module, so you'll be able to follow along each and every instruction that is given as well. Now, without a delay, let's get started. Now, to be able to view the different levels of protein structures, we need to first of all install a library into our notebook. We'll install Biopython. Biopython includes different tools for biological computation. We'll use it to fetch the protein sequences from NCBI and also read the sequence files. And the quiet here suppresses any extra installer messages. So once you have installed the Biopython library, the next step is to pull out a real protein sequence from NCBI. I'll set a contact email here as NCBI requires it. Here we'll define a tiny helper as well that will help us fetch the session ID, and then we can print what we get. So let me explain to you the line of codes as given here one by one. This line of code will help you import the two important modules which are required. One is SeqIO and another one is Entrez. The SeqIO lets us read and write the sequence files like FASTA, and Entrez gives us the access to the NCBI's database through Python. The next thing that you have to do is mention your email ID. NCBI requires you to set your email whenever you fetch the data, and this helps them contact you if needed. Then you need to define a function. So we'll define here a function that takes a protein accession ID. The term entrez.efetch here helps us fetch the data from the protein database using the accession ID that we will give to it. And also we'll set read type as FASTA and read mode as text for it to be able to ask for a FASTA file, and the text simply means that the FASTA file should be in the text mode. And the SeqIO.read function will help us read the FASTA file into a Python object. And finally, using handle close, we'll close the network connection. And SeqRecord will return us the sequence which was extracted through this accession ID, the sequence in the FASTA format, right? Now, the next thing is we'll call out the accession that we want to fetch the sequence for from the NCBI. And then we print three things. The first one is we will print the sequence ID, the sequence length, and the sequence itself. So we call out the accession ID that we want to fetch, save it into accession, and then we fetch the protein sequence from this accession ID and then save it into protein seq record, and then finally extract the sequence ID from the protein seq record, extract the sequence length, that is how many number of amino acids are present in that protein sequence from the protein seq record again, and from the same data, we also extract the sequence itself. So we can see that the sequence ID was this, sequence length is 1273 amino acids followed by the actual sequence of the amino acids in the protein. So that is how you can fetch a protein sequence from NCBI. Now, to be able to view the secondary structures, the protein secondary structures, we would need a library called as Py3D-mol. Now, what Py3D-mol does, it's basically a Python wrapper for a Web3D viewer, and we'll use it to render the protein structures directly into the notebook. So once you have installed Py3D-mol, the next thing is to import the Py3D-mol to be able to use it. We'll then define a function here, which will help us create a reusable function that takes the PDB ID and then helps us visualize it as a structure. So in this function, what we would do is we'll create this function called the PDB ID, where we will fetch the structure directly from the protein data bank. And then we set the style. In what style do we want to visualize the structures which get extracted from the protein data bank directly? So we set the display style as cartoon, where we want to look at the protein structure as ribbons. So we'll see ribbons which highlights helices and the sheets. And the spectrum colors the residues from blue as N terminus to red for C terminus. So we set the color as spectrum, and we set the kind of visualization as cartoon, and then we finally view the protein structure to render the 3D view of that specific protein from PDB. So once we have installed Py3D-mol, once we have defined this function to be able to visualize a protein by calling out its accession ID or unique ID from PDB, we'll then create these different levels of protein structures: alpha helices, beta sheets, beta turns, and also we'll have a look at the tertiary and the quaternary structures. So what you see right now on your screen is an alpha helix. To load this protein, we call out the protein IZTA. That's an alpha helical segment. And what you'll see here is a coiled spiral that is stabilized by hydrogen bonds along the backbone between the residue I and residue I plus 4. And the side chains point outward like bristles. We can also visualize the beta sheets. The code remains the same. It's just the protein ID type that gets changed. Here you will see the beta sheets which are connected by the loops. The sheets are flat and they are rigid. The loops let the chain bend and often host the turns. The beta turns are 4 residues long and commonly include the glycine and the protein. So what you see right now on the screen is a clean beta sheet snapshot. Now let's have a look at one of the beta turns as well, where we'll simply call out this specific protein 1LE1. And what you can see right here is a beta turn. You can zoom in and zoom out simply using the drag of your mouse. Then comes the tertiary structure. The tertiary structure is a full 3D fold of one polypeptide, which is stabilized by hydrogen bonds, ionic bonds, which are salt bridges, the hydrophobic clustering in the core, and the disulfide bridges between the cysteines. The common building blocks that you'll meet in the tertiary structures include the beta-alpha beta motif and the TIM barrel, which includes 8 beta strands surrounded by the 8 helices used by many enzymes. And then you have the quaternary structures. The quaternary structures means multiple subunits which are assembled together. For example, hemoglobin, which is alpha 2 beta 2 and uses a prosthetic group called heme, which binds to the oxygen. Now, to be able to create a quaternary structure, what we will do is we will call the function view from the library Py3D-mol, and we mention which specific protein do we want to work with, which is 3BCQ, which is our query here. And after this specific protein has been fetched from PDB, we set the style. We want to view it as a cartoon-like protein, and we set the color as spectrum. And if you want to add any other style, we can do that. Here we add the heme groups in a stick representation. The heme here selects the heme ligand, and we set the color scheme as JMOL, which colors the atoms by the elements. As nitrogen will be given a blue color and oxygen would be given a red color. So this is quaternary assembly plus the prosthetic group. And here each subunit cradles a heme, and the iron in the heme binds to the oxygen. So that was how you can easily visualize the different levels of the protein structure. You also have here a tutorial recorded by Dr. Sitharama Joy. So if you click on it, you can go through this tutorial to go through the different protein structure visualizations in PyMOL. So until here, you learned how to fetch a real sequence, that's a primary structure. Then you visualize the helices, sheets, and turns, that's secondary. And then we discussed about tertiary stabilizing forces and explored a quaternary complex with a cofactor. After you have completed the Python scripts, and once you scroll down, you'll see a section which says tutorials. So in all these submodules that we would be going through, you will find these tutorials, which includes activities for you to perform. And these activities are different sorts of practical hands-on, which will help you understand different concepts altogether. Now, before we really begin with these tutorials, you need to understand how the PyMOL in this specific interface of Jupyter Notebook in the VM would really work. So in the previous lecture, we did discuss how you can open up a PyMOL notebook and then how you can launch the PyMOL. Now, for us to go through these practicals easily, what I have done is I've just pushed the tab of PyMOL onto my other screen. So I would just work around with PyMOL and the code blocks from here, because I need to look at the code blocks, the command line code blocks, and then run it into Python. So it will be easy for me to play around with the PyMOL, looking at the different code blocks that I need to run in the command line of the PyMOL. Now, before we really get started, you need to understand how the different data files can be called in PyMOL and where our results will be saved for the codes that we will execute in this notebook. So if you look at the PyMOL interface right now, and if you go to this file option right here, click on open, you have a folder called as config. You need to click on the bucket folder, and then you'll find the structural biology and drug discovery course. Once you double click on it, you have all your submodules placed here. Currently, we are working with submodule 1. So if you click on it, you can see the data files, the PML script folders right here. To extract any of these data files that we would like to work with, you can import these data files from here. And when we will run the PML scripts in our notebook, the PML scripts would get saved in this folder right here. We can call these PML scripts in the PyMOL interface and then look at the result that the PML script gives us. So the first thing first, you have to make sure that your working directory has been set to structural biology and drug discovery, submodule 1 protein structure, as this is the submodule 1 that we are working with. So in case you have not checked your working directory yet, you just check your directory. And the next thing that you have to do is create a folder called the PML script. I can already see the folder of PML scripts here because I executed these codes before as well. And you can only see this folder once you have executed this line of code. Now, why do we really want to work with the PML scripts, and what are these PML scripts all about? These PML scripts are text files of the PyMOL commands that helps us automate the protein visualization and the analysis, and will help us run them to ensure reproducibility, and will help us save time and also make workflows easy to share. So let's say you have some specific number of tasks that you have to follow again, but for different structures. In that case, you can have a PML script created. All you have to do is load the different protein structures that you want to work with, and then execute the PML script to get the kind of protein visualization that you want to look at in PyMOL. So once we go through these PML scripts, it will be more easy for you to understand what these PML scripts really do. And once you have created the PML script folder, the next thing is to enable the write access from the GUI to the subfolders. This specific line of code block would not be accessible by you, as you cannot make any changes in the folder that we are currently working with. But it will be helpful to you if you create your own project and if you're supposed to work in a specific directory that you created. So CH mode here basically avoids the permission denied surprises in the classroom clouds. Now let's get started with these different activities. So the first activity will help us understand how the helix holds together. It's because of the interactions between the ith and the I plus fourth atom, which creates a backbone of the hydrogen bonds. So let's expose these hydrogen bonds and see what these hydrogen bonds look like. Now I would go to the PyMOL interface and load the data file called helix.pdb. So I'll go to the data folder, select helix.pdb, and then click on open. I'll maximize the screen more. So now what I see on my screen is a helical structure. Now, to view the hydrogen bond between the atoms in this helical structure, I will first of all hide the cartoon structure of this structure that we have with us in PyMOL and just view the lines. So I type hide cartoon and show lines in the command line. So I'm just following these steps which are mentioned here. I loaded the data and then I just hit the cartoon, type visualization, and then showed the visual as the lines. The next thing that I need to do is I'll display the sequence of the protein that I'm currently working with. Now here in PyMOL, you have these different options that you can work with. A is action, S is show, H is hide, L is label, C is color. Now, because I want to look at the polar contacts or the hydrogen bonds between the atoms, what I will do is I'll go to this action button right here and then go to the find option, polar contacts, and then select within selection. I've just followed the steps which are given here. And then I can label the residues as well. To label the residues, I can go to the label option right here and then click on residues. Once I do that, the residues will be labeled. All right, all these residues have now been labeled. So this was a simple practical where you can view the hydrogen bonds between these different amino acids, and you can also label the residues or the amino acids in the protein. You can use the right button of the mouse to zoom in and zoom out your protein structure. So just click on the right button and then move your mouse. And you can also use the drag option. So if you drag upwards, you'll be able to visualize the entire protein structure. So that was our simple activity one. Now for this activity one, we have also written a PML script. So you see, I went through these different steps and I had to use the command line to run some of the commands. And then I also use these options right here to be able to visualize the polar contacts and then label the residues. But what if I have different structures that I want to work with and I want to do the same thing? In that case, I can write a PML script. And in these PML scripts, I have mentioned the different steps that I followed here. So what I did, I loaded the structure, then I hit the cartoon type representation, showed it as lines, and then I displayed the sequence, selected the helical region, selected the polar contacts within the selection, and then labeled the residues. So now let me show you how this PML script will really work. Now to execute a PML script, you go to the code cell and do a shift enter. Once you do that, it says the PML script helixanalysis.pml has been updated and saved where in your directory. So I would reinitialize this PyMOL interface, and then let us go to our directory. But this time we'll go to the PML scripts folder and then look for the helixanalysis.pml, and I'll open it up. Now, what you can see here are the different residues labeled in the line-like structure of the protein. And if you want to visualize the polar contacts or the hydrogen bonds, or what I can do is simply go to action, find polar contacts within selection, and I'll have them up. So that is how you can automate a specific activity or a task in PyMOL. Now let's go to the next activity, which is to visualize the hydrogen bonding pattern in a beta sheet structure. The beta sheets are rigid. So where does that rigidity come from? It comes from a ladder of interstrand hydrogen bonds. Now let's draw them using PyMOL. I'll reinitialize this. And now what I should do is I will fetch a specific protein that I want to work with, which is 5GV0. Okay. In this activity, it's mentioned here that I want to fetch this 5GV0 protein. I'll click on enter. Then it says go to display and click on sequence. I did that. The next thing it says is that this time I have to hide everything and not the scene. And then it says that I have to select the beta sheet region and then hide everything but not the scene. To select the beta sheet portion, what I can really do is I can write a command saying select the beta sheet as the secondary structure. I can select that. So it says 779 atoms selected. And then I can say hide everything all. So I'll hide everything. And then I'll only look at the beta sheets. So just show the beta sheet as cartoon. Beta sheet as cartoon. Next, I'll specify the distance. So I select the beta sheet and specify the distance to expand to only 3 and mode 2. All right. So I've selected the beta sheets only and I've hidden everything else. I can visualize it as cartoon. So I can say set cartoon flat sheets. And I can also zoom in the beta sheet. You need to take care of the capitalization. So I'll zoom all of it. We can also label the beta sheet and I can name the carbon atoms. Right? So I have them labeled now and I can also set the label size if I want. 218, let us say. This is a better representation where I have the residues of the beta sheet now labeled. And then follows the same steps where I can really look at the polar contacts. I can look at the hydrogen bonding of these different residues here. So for that, I'll go to action, find polar contacts within selection, and I can look at the bonding. Though it's too cluttered, but that's the way you can look at the polar contacts within the residues. So that was your next activity. Now let's go to the activity 3, where we'll spot the beta turn in hemoglobin. So what is a beta turn? It's a tight 4 residue reversal often stabilized by Ni to I plus third contact. So let's isolate one of these beta turns and have a look at it. So what I'll do is I'll reinitialize all of it again. Now let us quickly fetch 3BCQ. You just have to follow along these instructions that have been given here. I'll look at the chains B, C, D. So because this is a hemoglobin that we are working with, we have 4 chains. I'll remove the chains B, C, and D. Next, I will remove the residues 1 to 30. I can also remove more residues, and then I will select these specific residues that have been mentioned here as 50, 51, 52, and 53. So I select these residues and then I'll show them as sticks. So I can look at these residues as sticks. So the beta turn has been highlighted here as sticks, just as you can see here. I can also label this so that I can see which residues are a part of it. I've labeled it now. I can also give it a different color. Let us say I can color it yellow. All right. So you can play around and then see how would you like to change the color or maybe the shape of it. But this is what the beta turn looks like. And we have also labeled the residues which are a part of the beta turn. We also have the PML script for this. So you can run the PML script. It includes the same steps where you go to file and then open, and then you open the activity 3 as the specific PML script has been saved as activity 3. And when you open it, just make sure that you reinitialize and open it. So once you open the activity 3, you can see the same protein structure with the highlighted beta turn. All right. If you want to see what PML script does exclusively, you can reinitialize your PyMOL and then have a look at it. So that was your activity 3, quick enough. Now let's move to the activity 4. Activity 4 is about disulfide bonds. So here we'll see how disulfide bonds stabilize 2 chains of insulin. Disulfide bonds are covalent zippers that lock the folds and connect the chains. And insulin is a great example to look at the disulfide bonds. So we will first of all fetch this protein. Fetch 3I3Z. And then we will go to show disulfides and then click on lines. Okay. This will help us look at these disulfide bonds as the lines. We can also zoom in a bit to look at these lines. We can also label them. We can also rotate them and understand where on which residues these bonds are present. So that's how you can look at the disulfide bond. Now let's move on to our 5th activity where we'll try to understand how the heme group binds to the pocket in hemoglobin. Now for this, you have the command line codes as well that you can execute in the command line of PyMOL, or you can simply run this PML script. I'll run this PML script to quickly see what kind of result do we get from this PML script, and I'll leave this command line codes for you to practice. So first of all, I have to run this. It says that the PML script has been saved as activity 5. So I'll go to open activity 5, click on open. Now this is what I can see where I can look at one of these parts of the hemoglobin with the heme group. where chain A can be seen as the green helices and this ferrous hair can be seen as a small pink or a red sphere. You can also click on the specific video example that has been put together where we have the detailed tutorial on how you can really look at the interaction types as well. And with that, we have completed submodule 1.1. And with that, we have completed submodule 1.1. Now in the next recording, we will look at submodule 1.2.